Hello everyone. Today we are going to see the topic staining and what are the types of staining. So coming to the staining, which means stains, it is a substance that adheres to the cell which gives the cell the color. So in this meaning, cell indicates the type of microorganism. So it is used to enhance and contrast the biological specimen at the microscopic level. So when we want to view some of the organisms, some of the microorganism in microscope, we need to stain that. So the bacterial cytoplasm is very transparent, so it is very difficult to see under the naked eye. So it is necessary to stain it before they can be viewed under the microscope. So we need to necessary to stain in some of the substances like dyes and staining colors. So stains and dyes are used to highlight that specimen and which gives the color to the microorganisms. So different strains have different affinities for different organisms. So coming to the biological stains, there are some of the biological stains used in the microbiology which is mentioned in this PPT. So first one is acridine orange, which is orange in color. It is mainly used to detect the nucleic acids. So nucleic acids is nothing but the DNA and RNA molecules, which is the very major genetic material. And it is acridine orange is mainly used to analyze the DNA molecules. And second one is Kumasi blue, and it is mainly used to stain the proteins. So proteins are the molecules, so proteins does not have any color at all. So when we used this Kumasi blue, the proteins will stained as blue color. And coming to the third one, crystal violet. So this crystal violet stain is very important in the gram staining, and this violet color gives the bacterial cell wall appears in the purple color. So when we see the bacterial cells in the microscope, the bacterial cell are appear in the purple color. And coming to the ethidium bromide, and it is the red color fluorescent stain. It is also mainly used to analyze the DNA molecules in the agarose gel electrophoresis. So when we run the agarose gel electrophoresis to visualize the DNA molecules, we need to add the ethidium bromide dye to visualize the DNA molecules. Since ethidium bromide is very carcinogenic, we need to add a minor ml of that reagent into the DNA. And next one is iodine. So it acts as mordant in gram staining. So crystal violet and iodine, which combines to forms the crystal violet iodine complex in the gram staining. And next one is methylene blue. So methylene blue is nothing but it is used to stain the animal cells. And coming to the safranin, and this saffron is also regarded as the counter stain for both the gram staining and endospore staining. So these are the stains or dyes which is most widely used in the staining processes, which will vary according to the type of the staining. So next we need to go for the types of staining. So coming to the types of staining, this simple staining is most widely used staining technique, which uses only one dye to determine the microorganism size, morphology and the arrangement. So a very good example is crystal violet, methylene blue and safranin. So if we add crystal violet, we don't want to need to add any other colors, because crystal violet itself colors all the cells. That is I used to mention only one dye. So if suppose crystal violet is used, so all the cells are appear in violet color. If suppose methylene blue is used, all the cells are appears in blue color. If the safranin dye alone is used, all the cells are appeared in red color. So this is the form of simple staining. You could see the image in the slide, which is coccus and coccus or spherical shape, or rod or bacilli shape. So this is the overall cells of the color appears in a single color. Next, coming to the gram staining, and this gram staining is very important to differentiate the organism. So this gram staining is mainly used to differentiate the gram-positive bacteria and gram-negative bacteria. So the one common difference between the gram-positive bacteria and gram-negative bacteria is that based on its cell wall properties. So gram-positive bacteria has very thick peptidoglycan layer in the cell wall, and gram-negative bacteria has very thin layer of peptidoglycan layer in the cell wall. And moreover, the color also different in gram-positive and gram-negative. You could see the diagrammatic representation of the gram-positive and gram-negative in slide. You could see the gram-positive bacteria appears as purple color, and gram-negative bacteria appears as red or pink color. So this is the major difference. Since gram-positive bacteria retains the crystal violet color so that it appears as violet in color, and gram-negative bacteria retains the saffron in color, which is red color, and it appears as red or pink color. So you could see both the shapes in both the bacteria. So you could see the cocci shape, that is nothing but the round shape, and you could see the rod shape, that is nothing but the bacilli shape. So you have both the gram-positive cocci and gram-positive bacilli, and also you have gram-negative cocci and gram-negative bacilli. Okay, so this is about the gram staining. And coming to the Ziehl-Neelsen staining. So you see, Ziehl-Neelsen staining is also called as acid-fast staining. So as like gram staining, Ziehl-Neelsen staining is also used to differentiate the acid-fast bacterium and non-acid-fast bacterium. Okay, so acid-fast bacterium, in the sense, a very good example for acid-fast bacteria is Mycobacterium tuberculosis. And the second point, you could see it is developed to detect the bacterial species that cause tuberculosis. So you need to know tuberculosis causative agent is Mycobacterium tuberculosis. So this Mycobacterium tuberculosis is a very good example for acid-fast bacteria. So in this, there also the morphological shape and structure will vary from one to another, and acid-fast bacteria will appear as red color and non-acid-fast bacteria will appear as blue color. So today we are going to see about this acid-fast bacteria in detail. Before that, we have another types also.